Welcome my dear students. I'll be talking about minerals in this section. So what are minerals? You know that what are minerals. So there are what are called as major minerals and the minor minerals or the trace elements. Major minerals daily requirement is more than 100 milligrams and trace elements daily requirement is supposed to be less than 100 milligrams. This is a crude classification which is being used. The major involve calcium, phosphorus, magnesium, sodium, chloride, sulfur, fluoride. And the trace elements include iron, iodine, copper, manganese, zinc, molybdenum and selenium. So out of all this, calcium I have already dealt with along with vitamin D, calcium and phosphorus we have already seen how they are metabolized. Now magnesium and all have got minor roles within the body. Mainly magnesium is involved wherever ATP is involved, magnesium is involved because of its +2 charge. It combines with all the kinases etc. Sodium, chloride, all these will be taught in different other sections, hypernatremia, hypochloremia, all other subjects deal with it. So I am going to focus my section on two important things. One is based on two important things. One, that it may not be dealt with in any other subject. Second, it is very important that even in this subject you have to learn about it. So based on this, let me start with the first mineral that I am going to talk to you about, and that is iron. So iron metabolism. Iron, the total body iron is around 3 to 5 grams is there. So if you look at the distribution of iron within the body, transport form around 0.05 to 0.18 milligram percent. Whatever has been absorbed contributes to the transport form in the plasma, and then this may be either used by different tissues or it may be stored. It may be stored iron in the liver, spleen, bone marrow, etc. 20 to 30% is stored iron. It will be used in muscles for the formation of myoglobin. It will be used by hemopoietic organs, especially for the formation of hemoglobin. Nearly 70% of iron is distributed. Total body iron is present in hemoglobin, and functional tissue iron, that is for example mitochondria contain cytochromes, and these cytochromes contain iron sulfur compounds. There are different enzymes which are present in different tissues. Functional tissue iron, or what we call as parenchymal iron, is around 5%. So all these are different ways in which iron is distributed in the body. Whatever is not absorbed is going to be excreted from the body. So let us look further. What are the functions of the iron, physiological functions of iron? Whatever functions are there, it is got because of the different proteins in which or different enzymes in which iron is present, and that is the function of iron. So it is present in hemoglobin, so transport of oxygen. It is present in myoglobin, so storage of oxygen. Cytochrome oxidase is present in the electron transport chain, thereby it helps in the production of ATP. It is present in the electron transport chain itself, like cytochrome B, C1C, succinate dehydrogenase, complex 3, all of them contain iron sulfur center, thereby it is responsible in the whole transport of electron transport chain and production of energy. Cytochrome P450, that itself has its own effects, like it is responsible for detoxification of drugs. So iron thereby helps in detoxification of drugs. Catalase, peroxidase, again these enzymes are responsible for causing different hydrogen peroxide is removed. So in these cases again they are heme containing enzymes, and thereby iron is responsible for all these functions. Nitric oxide synthase is also one of the iron containing enzymes. Nitric oxide is a vasodilator. Nitric oxide has its own different functions, thereby iron is required for the functioning of nitric oxide, for the formation of nitric oxide. Xanthine oxidase is also an iron containing compound, iron containing enzyme, so required for the degradation of purines. Aconitase, TCA cycle, phenylalanine hydroxylase, amino acid metabolism. Transferrin is the transport form of iron, and transferrin is also considered as one of the antioxidants. So again, and an acute phase protein, so transferrin, ferritin, hemosiderin. So thereby whatever functions of iron, you know that it is not just related to oxygen transport or storage of oxygen. It has got so many other functions, and one of the most important function is it is required by each and every cell within the body, by the mitochondria, by the electron transport chain, thereby for ATP production. What will happen if ATP is not produced? weakness. That is why in iron deficiency anemia we get weakness, because each and every cell, there is no iron and there is no production of ATP, production decreases, is affected, and that is the main reason for weakness, fatigue and all that is seen in iron deficiency anemia. If you look at the different proteins, you can able to tell what will be the different effects of iron deficiency. So it has a lot of functions, very important mineral within the body. So what are the sources? Sources, we have got different ways in which you can classify the sources. Community medicine are the people who tell you more about this, but I've just classified shown you the list. The RDA is also better told by community medicine people. For example, rich sources like all the non-vegetarian sources, liver, heart, kidney, spleen, etc. of different animals, meat, etc. are all good sources of iron, because in those sources, iron is in the form of heme, and heme iron is better absorbed. It doesn't need to be undergo any processes. So heme iron is always better than non-heme iron, and in plant sources it is always non-heme iron which is present, and thereby it is supposed to be less efficient when compared to heme iron. The absorption is supposed to be less of non-heme iron. Rich sources, even jaggery is considered as a rich source because of the way the production of jaggery is involved. It involves a lot of iron utensils which is supposed to give iron to the jaggery, and thereby it is responsible for the high rich content of iron in jaggery. But however, how much we eat, it all depends on that. What is the intake of iron, how much jaggery can you eat. But compared to sugar, jaggery is better. Good sources like egg yolk, fish, nuts, figs, dates, beans, spinach, apples, bananas. Poor source, milk is a very poor source of iron. Wheat flour, polished rice, potatoes, etc. Human milk is a very low source of iron, and that's why post-weaning it is recommended that the baby can be given once weaning starts, iron-rich foods should be given to the baby because of this. And it is sufficient at least for during the period of feeding, but later on the supplementation, it's not supplementation, extra foods need to be given. RDA, 20 milligrams of iron is required in adult Indian, 1 to 2 milligrams is absorbed. There are different categories of during pregnancy, during lactation, adult female, adult male, for adolescent, for children, everywhere we have different categories. Please always remember that the iron requirement of females is always more than that of the males. But once postmenopausal, the requirement of iron in both males and females is the same. Premenopausal, it is going to be drastically different. A female loses as much as 1 to 2 milligrams of iron per day if you consider the whole from menarche to menopause. During that time, she'll lose around 1 to 2 milligrams per day. 1 milligram, so requirement is more, especially because of the blood loss that is occurring. Now let us look at how it is being absorbed. You have got the intestinal lumen, the mucosal enterocyte and the plasma. So what happens is, in the whatever food content is there, whatever iron is there, it is going to be released. The protein bound iron is going to be released. Heme iron does not undergo so much of processes as of non-heme iron. My focus is going to be on the non-heme iron, the protein bound iron which is present in different forms in different vegetable sources. So FeOH3 may be there. This is broken down by HCl or any other acid to release ferric ions. Ferric ions have to be converted. There is no other choice within the body. They have to be converted to ferrous ions, Fe+2. Only and only then they can be absorbed. So this is one take-home message for MCQs. Only ferrous ions can be absorbed. So this conversion is done with the help of cysteine or ascorbic acid. Vitamin C is considered as one of the things. This ferrous ion then binds with a divalent metal transporter. This divalent metal transporter can help in the transport of iron into the mucosal cell. So once it is entered into the mucosal cell, this ferrous transporter with the help of DMT1, this mucosal cell, in the mucosal cell, this ferrous ion is reconverted back to ferric ions, Fe+3, and for this the enzyme is ferroxidase. Ferroxidase, once it is forming ferric ions, it combines within the enterocyte. There is a protein called as apoferritin. So all these ferric ions will combine with apoferritin to form ferritin. Ferritin is the storage form of iron. It gets stored in the intestinal mucosal cells also. That is when it depends upon the need of the body. So if the need is there, this ferritin will be acted upon ferrireductase to reform ferrous ion. If the need is not there, this ferrous ion is stored as ferritin. If need is there, sometimes directly it gets absorbed into the plasma. This peroxidase, ferrireductase, both of these need not be there. Depending upon the need of the body, Fe+2 is ferrous ions are absorbed into the plasma. In the plasma, what happens? This whatever ferrous ions are reconverted back to ferric ions. So peroxidase, what is important in all this? It is the ferrous ions which are going to be going in or out of the cell. When it is going to be stored or when it is going to be associated with a protein, it is always ferric. When it is going to be by itself, it is ferrous. Okay, so it is reconverted back to ferric ions. Peroxidase of ceruloplasmin, a copper-containing enzyme, and it forms Fe+3. This combines with apotransferrin. Apotransferrin is the transport form of iron. Apotransferrin to form transferrin. This is the iron that is bound to transferrin. This transferrin will take that iron to different tissues. Wherever it is required, everywhere it transports. Transferrin transports iron. If required, it is stored in these tissues also. Once it finishes its function, again the storage form is going to be ferritin itself in different tissues. So it reaches different tissues, liver, spleen, bone marrow, and then either it is used or stored in mainly liver, spleen, bone marrow. In all other tissues it may be used also. The storage mainly occurs in the liver, spleen and bone marrow. Storage stored as ferritin. So this is how iron is taken up by the different tissues. And there the functions, as I said to you, different enzymes and proteins require iron, and it is used for all those things. So let us look at more about absorption. Less than 10% is absorbed, mainly in the upper part of the duodenum. It is a ferrous form, it is absorbed. Ascorbic acid is required. Absorption is decreased, again an MCQ question, due to high phosphate, calcium or lead. Lead also inhibits it competes for iron, and it decreased absorption. Phytic acid, which is present in cereals and oxalates, interfere with the absorption. Gastric acid definitely favors absorption. Copper deficiency decreases absorption. Why? Ceruloplasmin is required for the transport, so copper deficiency can also decrease the absorption of iron. So the different factors affecting absorption. Now there is something called as the mucosal block theory. What do I mean by mucosal block? Now mucosal block is the way the body regulates the iron's absorption into the body. If the body needs iron, it is going to absorb iron. If the body does not need iron, it is not going to absorb iron. So what it will do? It will do it at two levels. One, it may be at the level of the divalent metal transporter. So what will happen? Whatever ferrous is there, which is in association with the divalent metal transporter, this DMT1, it may not be absorbed. It is just thrown out of the body. So at this level is one thing. The other thing is at the level of the mucosal cell. So as you've seen already, as I've shown you already, in the mucosal cell, it can be stored. So in the mucosal cell, it can be stored as ferritin. Now, whatever is absorbed, and whatever, when it is required, it will release that Fe+2 to the different tissues. Suppose this Fe+2 is not needed, the body does not require it, it will not release, the ferritin will not release this. So when the ferritin does not release this, what will happen in the intestinal mucosal cell? Whatever iron is absorbed will keep on getting stored as ferritin in the mucosal cell. So what? This mucosal cell are one of the cells which undergo rapid turnover. So every 5 days, every 6 days or every 10 days, this mucosal cell is going to be shed off and a new cell is going to come in its place. So when this mucosal cell is going to be shed off and being lost from the body, this ferritin, whatever has been stored, is also going to be lost from the body. So that is known as the mucosal block theory. So at the level of absorption itself, how much of iron, whatever the body requires, it will regulate at the level of absorption. So what determines all this? Maybe erythropoietin may be one of the factors which tells how much of it is to be stored as ferritin, how much has to be transported as transferrin. Erythropoietin is an hormone which is got from the kidney, and this may regulate it. So uptake depends on this shedding with the stored ferritin. Erythropoietin may act as a signal. Further, let me talk about the transport. As I said, apotransferrin plus ferric to form transferrin. Now this transferrin is taken up by tissues by binding to a specific receptor known as transferrin receptor. Each transferrin receptor can bind to 2 transferrin molecules at a time, and this receptor will recognize transferrin and will bind to 2 receptors. It is internalized. Once it is internalized, it releases that iron and then it is recirculated. It goes back to the plasma membrane. And the synthesis of this transferrin receptor, mRNA is also being regulated by the iron levels within the cell. If the iron levels are adequate, then this transferrin receptors are not going to be, the mRNA is going to get degraded. But if the iron levels are inadequate, the mRNA is going to be stabilized and it is going to see that more and more transferrin receptors are synthesized so that whatever is circulating, let the cell take it inside. So again, one more way by which the body is regulating the iron within each and every cell within the body. So the transferrin receptor mRNA stability and degradation is regulated by the iron status within the cell. Because of there is what are called as stem loop structures of the mRNA, which it can bind and can dissociate it from an iron regulatory protein, and the stability is going to be maintained. If adequate iron is there, the mRNA is degraded faster to prevent further uptake of iron. Now storage of iron, liver, spleen, intestinal cell. So whatever after it has been used, it forms ferritin. Apoferritin has 24 subunits and can take up to 4000 iron atoms per molecule. So, one important deficiency disorder associated with iron is iron deficiency anemia. We all know what is iron deficiency anemia, hypochromic microcytic anemia. I won't talk about that. I'll talk about more about the lab findings rather than the pathology or the peripheral smear findings. Okay. So what are the causes? Nutritional deficiency. Most common cause in India, the most common nutritional deficiency is iron deficiency anemia. Nutritional deficiency. Hookworm infestations. Yes, because they cause blood loss. Repeated pregnancies. Again, because of blood loss. Each pregnancy there is loss of iron from the body. So again, repeated pregnancy, iron deficiency anemia. Chronic blood loss. Maybe a peptic ulcer is there, bleeding ulcer is there, maybe melena is there, maybe some other reason because of chronic blood loss. Again, it can lead to iron deficiency anemia. Nephrotic syndrome. In nephrotic syndrome, there is loss of hemoglobin, haptoglobin or hemopexin. Remember, all these are involved in the conserving iron. Hemoglobin, minute RBC is lost, this hemoglobin is going to bind with haptoglobin to prevent its loss from the kidneys. Minute heme is released, heme will bind to hemopexin and this also prevents the loss of heme from the body. Iron is conserved. Iron is considered as a one-way element. Whatever is absorbed, the body tries its maximum to conserve it by it does not allow the kidneys also to excrete out iron, but in nephrotic syndrome, these may be excreted. There may be lack of absorption, maybe gastrectomy, you require acid for the conversion of ferric to ferrous. That is not happening. Gastrectomy might have occurred and that has led to iron deficiency anemia. Lead poisoning is again one more cause for iron deficiency anemia, whereby because lead inhibits so many different enzymes involved in both the absorption and further metabolism of even hemoglobin, and synthesis of heme is also inhibited by lead. So let's look at the lab findings. As I said, peripheral smear, I won't go into the peripheral smear portion of iron deficiency anemia, which will be more mainly dealt with pathology and physiology. I'll just talk to you about the lab findings, biochemical lab findings. Now, can we measure iron? Yes, serum iron can be measured and it is 120 micrograms per dl is the normal value. There is a concept called as serum total iron binding capacity, TIBC. What does it mean? It shows how much of transferrin, how much iron can it bind. So transferrin is a transport protein. As I said, it can bind to iron. We look at the total iron binding capacity. There is a mechanism by which we can do it. Now the normal is 400 micrograms per dl. And if there is iron deficiency anemia, that means the iron binding capacity is increased. Means all these transferrin are there which are empty. There is no iron. So it increases the total iron binding capacity is increased in iron deficiency anemia. Transferrin, can we measure it? Yes, serum transferrin levels also can be measured. It is around 250 milligrams per dl, and this is also increased in iron deficiency anemia. The body, in a response to increase the transferrin, whatever iron is there, let me transport it. So it increases the amount of transferrin also in iron deficiency anemia. But this transferrin is also increased because it is an acute phase protein, and so it can be increased in other conditions also. So it does not give a good reliable indicator of iron deficiency anemia. And nowadays there is what is called as soluble transferrin receptor levels, STR, and this is also increased in iron deficiency anemia. So iron toxicity can lead to hemosiderosis, where hemosiderin pigments are seen in liver and spleen. The causes are many, including primary Bantu siderosis or hemochromatosis, etc. More will be dealt with in pathology. I'll talk about one more mineral, that is copper. You already know about it. 10% of copper is absorbed. Whole blood contains around 100 micrograms of copper, and this is distributed in RBC erythropoietin, and it is mainly required for ceruloplasmin. It is also transported bound with albumin. Copper containing enzyme, this is the list. You can pause at this moment and look at the different copper containing enzymes. One important one is cytochrome oxidase. Again, it is required for ATP synthesis. So main functions: incorporation of iron into hemoglobin, iron absorption. Tyrosine is another important because it is required for pigmentation. Collagen and elastin are another two which require, because of the enzymes again, require copper. Wilson's disease is due to decreased ceruloplasmin, where there is a defect in the gene for copper binding ATPase in the cell, which is required for excretion of copper into bile, and copper deposits are seen in the brain and the liver. Hepatolenticular degeneration. There is what is called as Menkes kinky hair syndrome. Again, copper absorption cannot be transported, so absence of intracellular copper binding ATPase. One last thing I'll talk about is zinc. Zinc is present in a lot of proteins, and more than 300 enzymes require zinc. So extracellular insulin is stored along with zinc, and extracellular SOD contains zinc. So zinc and copper compete for absorption, and zinc is used in the treatment of Wilson's disease. Deficiency can lead to what is called as acrodermatitis enteropathica. You can read about this. What are the functions? Selenium is the last mineral that I am talking about. It is present in glutathione peroxidase, antioxidants, deiodinase, so which is responsible for thyroid hormones, and so selenium deficiency can lead to hypothyroidism. It is also an antioxidant which is used along with vitamin E. I have told more about selenium already along with vitamin E. Kishan's disease is one disease associated with MCQ point of view I am talking. It is a province in China where the soil is deficient in selenium. It is characterized by myocardial necrosis, cardiac arrhythmias, and cardiac enlargements. So with this we come to the end of different minerals. Again, I have told to you, I have chosen these minerals, which ones to tell, based on the fact that they may not be told in other subjects, and its importance. Iron is important, that you know about iron, whichever subject you are learning. So hence iron I have included. The remaining minerals have got different other functions, like fluoride and all are taught in community medicines. You will learn more about it in different other years also. So with this we come to the end of minerals. Thank you.